Okay. Hi, everybody. I hope that you are all doing well. At this point, you should have been able to see on YouTube all of the past lectures except for lecture 3, which I still need to finish doing technical stuff with. But the rest of them are all up. So that's the deal with that. I will say this again at the end today. But I want you to be really aware, Monday and Wednesday of next week, we're going to be talking about generation of antibody diversity. For many students, that ends up being the hardest thing we talk about in the course. Please, all the pleases. If you do not usually do this, please do the reading in the textbook before coming to class for those days, because it will be very helpful for you to have heard the words, have seen kind of those topics before you first hear them from me, instead of being like, why is she now talking another language? What's going on? If you've at least seen it before, that will definitely help. If you look going forward in the syllabus, you will note that you have two problem sets to do the week after next. One on Monday, one on Friday. So next week we've got generation of antibody diversity on Monday and Wednesday, and then we've got immunology techniques on Friday. There's a problem set due the following Monday that's really on antibody diversity, and then the following Friday that's about techniques. I have it set that way so that you have time to come to office hours potentially for either of those, if you need to, before they're due. I didn't want to, complete talking about diversity on Wednesday and then have you need a problem set on Friday and feel like you didn't have time to ask questions. But please realize I set up that timing for you to have time to ask questions. So that only works if you start working on things a little bit early so you can actually come ask the questions. Also, I have both of those problem sets set so that I can turn them around, though the technique one will be tricky, before exam 1, because some of the questions on those are going to be related to things you're going to see on exam 1, and they're things I know people sort of need practice with in the questions. So know that that's kind of the point of those, is that they are things to help you prep for exam 1. I would recommend working on them earlier rather than later so that you have time to come to office hours or ask questions as needed. They will be up this weekend if you want to work on them this weekend, even though we haven't done the stuff in class yet. Sure. So they'll be up this weekend, but please try to take a look at the material before class on Monday, at least in the book, so that you will have heard some of this stuff before I start talking about it on Monday. Today we're going to continue talking about antibody structure and function. We left off thinking about some of the general details of antibody structure, which you can see here. So you will notice that with our antibodies, we have those two identical heavy chains shown in green on the left, shown in yellow on my little stuffed antibody that I have here, and two identical light chains as well, yellow in the image, orange in my little stuffed antibody. I can see that there are two disulfide bonds holding together the heavy chains and one disulfide bond each holding a light chain to a heavy chain to give us this structure. And we've got this area that is the antigen binding region, which is on these two shorter arms, as well as this region that I called the constant region for you last time. If you look at this view of antibody structure, you will notice that you see the antibody drawn as being made up of a bunch of rectangles. And I often also will draw with those same rectangles being put in. Just as a thing, if you look at some of the old exams, one of the ways I had a question written in a past year, was I had a question where I had the rectangles drawn like this. And the same on the other side, but I'm lazy, so I'm not going to draw it right now. And I asked the students to tell me which things were covalently linked. So basically I wanted them to say, like, these two go together as the light chain. And these go together as the heavy chain. I thought that was pretty straightforward, but oh my gosh, did I get things. It was like I was getting all sorts of stuff. So just know that the point of if you see a past question like this, it's just to know, like, which of these rectangles sort of are part of the same protein. And those rectangles that I just drew on the board, as well as the rectangles you see here, are a very common way that immunologists will be drawing antibodies. And that's because those rectangles each stand for a particular protein fold or a particular type of domain. That domain was first described in the antibody. And so when it got named, it was named the Immunoglobulin domain. So each one of those rectangles that you see is an immunoglobulin domain. And this is the structure of the immunoglobulin domain. You will notice that the immunoglobulin domain is mostly made up of beta sheets. So, and you can see their direction here, which way they're going. They're basically anti-parallel beta sheets. And they are held together by a disulfide bond in the middle. Really, what this actually looks like in three-dimensional form is like a little sandwich with a toothpick in the middle, holding it together. And so here you can kind of see it more as a sandwich, more of the two flattened together. This is it, sort of all spread out. You can also note that there are some loops that connect the beta sheets. And so you can see those loops here. This protein, for example, starts here at this amino terminal and goes up, and you've got a beta sheet and then a loop, and a beta sheet and then a loop, and then a beta sheet and then a loop, and a beta sheet and then a loop. I'm going to be mentioning those loops in a little bit. So notice the loops now. You can actually see in the 3D structure of the immunoglobulin domain, those loops coming off of that little sandwichy part. And again, they're going to be important in a couple slides. This is just the view from your textbook of the immunoglobulin domains. And so you can see, in one FAB, perhaps one part of a light chain, there's, this little rectangular area, which is this immunoglobulin domain. And it's connected to this little rectangular area, which is this immunoglobulin domain. And so this is just sort of another way of showing you the same thing. This is the version that you'll see in your textbook. This is an important protein fold in biochemistry. It was first described in antibodies, which, if you remember, I told you antibodies are sometimes also known as immunoglobulins. So that's how it got the name immunoglobulin fold. But now we know it's in tons of proteins. Particularly, it's in a lot of super important proteins in the immune system. So I think about the immunoglobulin fold being an important fold in a lot of the proteins I see in the immune system. You can see that this textbook sort of draws these little half-circle-y things as a part of many of these different proteins. And that's how this textbook tends to draw an immunoglobulin fold, is with this little half-circle-y thing. There will be times when I will be drawing proteins on the board later in the semester where I will be drawing it as little half-circles. And those are also the immunoglobulin folds. So the point that I want to show you here is that they were first described in the immunoglobulin. Here is an immunoglobulin. But a lot of the super important proteins we're going to see over the course of the semester all do have this same fold. So here you can see, again, that same antibody structure that we have seen. There are some short segments that are not IG fold, which are really kind of these short little connecting regions known as the Hinge region. So for basically the next few slides, I'm going to be telling you, like, what's the purpose of different regions. So to start with, I'm going to tell you what the purpose of the Hinge region is. because it's kind of the littlest one that we don't say as much about most of the time. And the hinge region is really key for flexibility of antibodies. So any particular antibody molecule has a lot of flexibility at the hinge. And so antibody molecules have the ability to wave, so they can move their FABs closer together and further apart like this. They can also kind of wag the tail, which I'm not going to do. They can rotate around the hinge, so we can have the FAB arm at different angles, and you can see that they can even bend a bit there too. And so we do have a lot of flexibility that's coming from the hinge region, which is sort of the one not immunoglobulin fold region. We can also look at what we're seeing in some of the other portions. And first, I want to talk a little bit about the antigen binding portion. So we can think about this as being like the FAB or something like that. I already kind of previewed what I'm going to tell you on the next slide last time when I told you about the beta-mercaptoethanol reduction of the antibody, where the heavy and light chains got separated because we broke disulfide bonds. And what I told you then was that heavy chains by themselves or light chains by themselves didn't bind antigen. Another way of phrasing that is that you have to have the heavy chain and the light chain combined in order to bind antigen. And so the binding site for antigen is made up of sort of a combination of the heavy chain and the light chain that we have here. If we actually paired a different light chain with this heavy chain, we would have a different binding site and we would bind a different antigen. And so you can see this here. You can see our antigen binding to these antigen binding arms and making contact with both the heavy chain and the light chain. So we often talk about, well, I'm going to come back to this slide because there's a piece on here that is related to something later. But in fact, this is actually showing you the antigen. This is showing you the heavy and light chain. And what I want you to notice is that it is sort of like a contiguous surface of the antibody binding to the epitope. Also notice where the antigen is binding. Sometimes in the past I have had people draw an antibody and with an antigen bound to it on old exams, and I sometimes have people who put the antigen here, and that is sad. So remember, this is where the antigen is going to go. Here you can see just another version of this, that the heavy and light chain are coming together, making some kind of shape that is going to be complementary to the epitope. And so it's really the combination of the heavy and the light chain that are coming together to bind antigen. And so this is just showing you that same thing another way. When I was in undergrad, and my roommate was a political science major, and I was trying to explain immunology to her, I told her it was just shapes. I was actually just learning about the things you learn in preschool about shapes that match each other. And so it's the same thing. Heavy and light chain come together, they make a shape, and that matches the shape of some epitope. But to go in a little bit of additional detail, we can think about the specific immunoglobulin domains and some of the details of the immunoglobulin domains. Sometimes we refer to these immunoglobulin domains, just the one Ig domain at the very end of the heavy chain and the light chain. So there are 4 such Ig domains in this picture, all shown in red. We refer to those as variable regions, while we refer to the ones that are shown in blue as constant regions. So the variable region immunoglobulin domains are the ones that are making contact with the antibody. If you had to guess, I know this is going to be shocking. If we compared amino acid sequences among different parts of the antibody, the place where you would see the most variation is in the variable region. But we can actually do that kind of analysis where we look at variation in some more detail. And when we do that, we find that those variable regions are the most variable parts. But if we map how much variability, which is shown here on this y-axis, there is at different parts of the protein, we find that there are 3 parts that are extra variable. They are the most variable parts of the variable region. Those are sometimes referred to as the hypervariable regions. And so you can see that there are 3 hypervariable regions within the variable region here. If we actually then try to map those hypervariable regions onto the structure of the immunoglobulin domain, we find that those 3 hypervariable regions are the 3 little loops that are coming off the end of that variable region's IG domain. So for both the heavy chain and the light chain, the really variable part are like 3 little finger loops that are coming off like this. So there's 3 little loops on the heavy chain, 3 little loops on the light chain, on both sides. And so you can see that here. You can see them listed as HV1, HV2, HV3 because they are hypervariable loops. But if I go back to my earlier slide, which also showed them, there's another way we think about them. So this slide also showed those 3 loops. Here you can see they're kind of shown as like different colored areas. In this image on the right, you can see this sort of pinkish blob thing. Okay? That is the epitope. Here it's labeled as the antigen, but it's in fact the part of the antigen that is the epitope. So here you imagine that you are the epitope, and you are staring at the antibody. It's staring at the antibody, right? When that happens, it turns out that you are getting 3 contacts, 3 touches from the heavy chain and 3 touches from the light chain. You're getting 3 from the heavy chain's variable region. See how there's VH, variable of the heavy chain, variable of the heavy chain, variable of the heavy chain, and 3 touches from the light chain variable region. So those 3 little fingers that are coming off the heavy chain and the light chain are actually the thing that is touching the epitope. They are the thing that determines that shape that I showed you before. Specifically, they determine whether or not that antibody is complementary to that epitope, like whether they have the matching shapes. And so we often refer to those 3 loops as the complementarity determining regions, or the CDRs. And so you can see here they're listed as CDR1, CDR2, and CDR3, the complementarity determining region. And they are actually what's really touching the epitope. And so when we think about generation of antibody diversity, we are specifically going to care about how in the world we get diversity at those CDRs. And they are forming these little loops at the end of the variable region's immunoglobulin domain. And so you can see that's where the most of the diversity maps. Those are the little loops. They are actually the very tip of the antigen binding site that will be binding to the antigen. Does that make sense? So at this point, we've thought about what's going on with the antigen binding part of the antibody. But there's this other tail down here too. And so we need to think about what's going on with this tail. We often talk about this tail as being called the constant region, or the FC. Light chains do have a constant region. So you can see there are light chain constant regions. There are also heavy chain constant regions. The heavy chain constant region is bigger than the light chain constant region. We're going to say as much about light chain constant regions in like 2 weeks. Other than that, we really don't care. But we can think more about the constant region of the heavy chain. Again, usually talked about sometimes as the FC portion, or this part is the FC. Compared to the variable regions, get this, the constant region is more constant. But when I say it is constant, it doesn't mean there's actually only one kind. With the variable region, and we'll talk about this more next week, in some cases we can say that there are about 10 to the 16th different variable regions. So that's, what is that, billion, trillion, that's 10 quadrillion. So that's a pretty big number. For constant regions, at least for our purposes right now, there are 5, which is a bit less than 10 quadrillion. So pretty constant compared to the variable region. Those 5 are listed here. One of them is called M, and an antibody that uses the M constant region will be called IgM, Immunoglobulin M. One of them is D, one of them is G, one of them is E, one of them is A. You will notice here that we've got colors for the heavy chain constant region. The light chain's not involved. The light chain constant region is a whole other thing that we don't really care that much about except for in one place later. This might not make sense now, so I'm going to say it and just believe me, and we'll get to like exactly what I mean by it later. IgM is encoded by a gene called the mu gene, or c-mu. Mu is the Greek letter for M. IgD is encoded by the delta, C delta. IgG is encoded by C gamma. IgE is encoded by C epsilon. IgA is encoded by C alpha. And then in each case, it's the Greek letter that corresponds. For some of these, there are actually subtypes. So at some point, I will mention things like IgG1, IgG2, IgG3, IgG4. But really, at least right now, you need to know that there's IgG. I remember these with a mnemonic. You guys have learned about how an apple a day keeps the doctor away, right? So what I learned was that MDs give everyone apples. So that will give me my 5 different types of constant regions. The order of these will actually be important to us in future days, and it gives it to us in the right order too. We will refer to antibodies with these different constant regions as antibodies of different isotypes. And so we usually say that these are the 5 isotypes of the different antibodies. We can refer to other kinds of differences besides differences in isotypes. So some of those types of differences are shown here. So first of all, you can see another example of an isotype difference. So here we have an antibody that is an IgE antibody and an antibody that is an IgA antibody. And they're both from Sherry. I don't know who Sherry is. We can also say that there are antibodies that are different because they have the same constant region, but they have differences in their variable region. Really a lot of those. They don't always talk about those as much, but if we were to sort of use this terminology, we would call those antibodies of different idiotypes. Usually we just say they're different antibodies, but officially they are antibodies with different idiotypes, or they have idiotypic differences. We can also look at the same antibody against the same antigen, specifically against the same epitope, from two different people, from Sherry and John. Sherry and John are genetically not identical. Somewhere they might have had like a point mutation that changes like one amino acid in the protein. And so their antibodies might not be identical. Here we can see it because Sherry's is orange right there and John's is brown. And so we would call these antibodies, they're basically the same variable region, same isotype, but slight differences between individuals as having allotype differences. Allo is usually something that we use often in immunology for different between two members of the same species. Yeah.
When vaccinations are made, somebody has, let's say, like John, for instance, has the IgG that it works well against antigen B, and the vaccine has like what Sherry has that works with antigen A. Do those two antibodies like combine, or do they just work together?
So in a vaccine, we are giving you something to push your body into making antibodies. Usually we're not giving you just one epitope. We're giving you a whole antigen. And as a result, Sherry should have something that works against that antigen. John should have something that works against that antigen. It's possible they're different epitopes because Sherry and John are genetically different, but they should both have something that will hit an epitope within that antigen. So now we've seen our antibody, which is just a protein with 4 chains held together by some disulfide bonds. If you went out and asked people who didn't know anything about science about antibodies and what antibodies do, you would hear all sorts of things. Antibodies, sometimes people describe them as being like total superheroes. It's like, dude, where's the cape? Sometimes people talk about antibodies phagocytose things. It's a protein. How can a protein eat something? Or, we think about all of these amazing ways that this antibody kills a microbe. And now you're like, it's a protein made with 4 chains held together by disulfide bonds. How the heck does it get rid of anything? How does it do all of these great actions? So what we need to talk about for the rest of our time today is how exactly antibodies function in getting rid of microbes or in dealing with microbes. I'm going to talk through the specific effector functions of antibodies. There are 6 of them in kind of broad strokes right now. And then we're going to come back and think about some details of different isotypes. So like I said, there are 6 functions of antibodies. And we're going to go through and sort of define each of them. And you're going to like the first one. You're going to like some of them a lot. The first one is a particularly good one. Because it turns out the first one, you already learned this semester, so you don't even have to learn a new thing. And you got one down. So the first effector function of antibodies is that they can start the classical pathway of complement. They can fix complement. So one way that antibodies can deal with microbes is by starting the classical pathway of complement. So you know that one already. Easy. There's a second one that's also relatively easy, pretty straightforward based on some of the things that you know right now. And that is opsonization. So you've already heard the word opsonization this semester. You saw an example of complement performing opsonization. And so what does opsonization, just as a general term, mean? Yeah, Grace.
Isn't that where, like, you coat the thing in something really desirable and then it gets eaten?
So you're coating a microbe with something, and that's making it more likely to get phagocytosed, making it more likely to get eaten. In the case of complement doing opsonization, you're coating the microbe with complement. In the case of antibody opsonization, you're coating the microbe with antibodies. And so we can have a microbe getting coated with antibodies, and that can induce phagocytosis of that microbe. Just like coating it with complement could also potentially have induced phagocytosis. When we saw opsonization with complement, one thing it required was a cell to do the phagocytosis. But it required one other thing. There was one other important thing when we saw complement opsonization. What was that one other thing? Is there anything about the cell that the cell had to be able to do or the cell had to have? Yeah, Joel.
They had to have something in place. I think it was like a receptor or something that disallows complement from binding to like their own cells.
So our cells do have complement inhibitors, but they also have complement receptors. They have to have something to see the complement that got coated on there and do the phagocytosis. Here you can see some receptors. And in the case of complement, we needed a complement receptor to recognize the complement that got stuck to stuff. In the case of antibodies, we need a receptor, but it's not a complement receptor in this case. If you notice the antibody getting recognized, which portion of the antibody is being recognized here? Yeah. Constant region. What was another term I used when I mentioned the constant region? Yeah. I mentioned the FC region. So we need an FC receptor. And so opsonization by antibodies involves cells that have FC receptors binding to the FC portion of the antibody and doing phagocytosis. And there are actually many kinds of FC receptors. So here you can see this one says like FC mu, it binds IgM. And there's one that's like FC gamma and it binds IgG. At this point, you just care FC receptor. But there are, in fact, lots of different FC receptors that act in slightly different ways. So those are 2 of our 6 functions. Complement, opsonization. Pretty much stuff you get. The third one is actually relatively simple. And it's, to be honest with you, the one that I think about the most because I think about viruses a lot, and it's sort of the virologist's favorite one. So oftentimes this is one, especially during the COVID pandemic, this is the one everybody talked about all the time. Right or wrong. This is the one everyone talked about all the time. And so this function is called neutralization. So in fact, in studies when people were looking at immune responses to SARS-CoV-2 or vaccines, they didn't just measure were there antibodies. They measured were there neutralizing antibodies, meaning were there antibodies that had the ability to do this function. In order to understand neutralization, again, we have to think carefully about some general ideas of how a virus is infecting a cell. Not super complex here. Here's a virus. It is interacting with some receptor on the surface of the cell, and that is allowing the virus into the cell. That interaction is key to allow the virus into the cell so the virus can replicate. When there is a neutralizing antibody, that antibody binds to the microbe, very commonly a virus, and physically blocks that virus or that microbe from interacting with a receptor so that the virus cannot infect cells. In some ways, I like to think about neutralization as being fancy steric hindrance. Basically, just the antibody binds to the virus and the virus can't reach the receptor anymore because the antibody's in the way. That's all. Not hard. Here you can see an example of a 3D structure of a virus. This is rhinovirus, one of the agents of the common cold. interacting with its receptor, and it's interacting kind of at this little blue site here. And in green, you can see what it looks like when there are antibodies coating this virus particle, and these antibodies can be neutralizing. You can see there are some antibodies covering up that spot. So now the receptor cannot bind to this virus. Sometimes the antibodies are binding to the exact same spot the receptor will bind to. Sometimes they're just binding nearby and physically getting in the way. Either of those counts. Either of those is totally fine as neutralization. If you remember, I told you about von Behring and Kitasato last time, and I told you that they were really lucky with the microbes that they chose, in that they got an answer of just serum was important. It turns out that the 2 microbes that they chose, well, really all 3 of the microbes they chose, cause disease because they make a toxin that floats around the bloodstream. Those toxins can bind to a receptor on the surface of a cell and harm the cell. See, it says it poisons the cell here. Antibodies are really good at neutralizing toxins too. So you can have the antibody bind up the toxin, so the toxin can never interact with the cell and never hurt the cell. And so in fact, they lucked out because they picked 2 bacteria that worked by toxins, and so antibody neutralization did everything for them. So it doesn't have to be viruses when we're thinking about neutralizing antibodies. It can be things other than viruses, particularly toxins. But most commonly when we're thinking about viruses, we talk about neutralizing antibodies, and we think about neutralizing antibodies a lot. So that's number 3. Again, none of these are like super rocket science. And now we're going to move to number 4. Number 4 is everyone's least favorite one. And the reason I say that is because number 4 always feels a little lame. Like you hear it and you're like, that's dumb. How does that count as one of the ways? It does have a lot of physiologic importance. But what we can see happening for one of the 4th methods of antibodies getting rid of or dealing with microbes is that we can see the formation of antigen-antibody complex. So sometimes we'll just talk about complex formation. Sometimes we even talk about this as something like precipitation or something like that. Basically, the idea here is that because your antibody has 2 identical arms that can bind antigen identically, and many antigens often have repetitive epitopes, you can get these complexes of many antibody molecules and many antigens coming together. And you can see a few different versions of those here. So you can see, this sort of linear one. You can see a little square one up there where you get multiple copies of antigen and multiple copies of antibodies all bound together. And basically, they make a clump. And they precipitate out of the blood. And here you can actually see a whole bunch of these antigen-antibody complexes precipitating out and forming a clump. And so you can imagine this is sort of, disabling the microbe, pulling the microbe out of circulation, something like that. Again, it feels lame. One reason why this one is actually really important is because sometimes these complexes can cause disease. There are a number of these that are very much involved in some diseases. So we are going to see them come up. If, for example, your kidneys get full of these little clumps, then suddenly your kidneys are not working anymore and you have nephritis, or something like that. And this can happen in a number of different body sites. In a perfect world, usually those antigen-antibody complexes become phagocytosed eventually. So don't worry that your entire body is totally full of them. They're probably getting phagocytosed and cleared, but we generally talk about complex formation or precipitation as one of the functions of antibodies for dealing with microbes. All right, so we're at 4 out of 6. Not too crazy, right? The 5th one is one that I always snicker when I talk about this one, because I'm going to tell it to you, and I feel like I'm going to tell it to you really clearly. And then we're going to see a part in like, I don't know, maybe like November-ish, where I'm going to tell you this term is going to come back, and I'm going to be like, what does this term mean? And you're going to be like, did we ever learn that? What? I never heard that word before. So I promise, I'm telling it to you right now. So one of the things that antibodies can do is they can lead to a function called ADCC. ADCC stands for antibody-dependent cellular cytotoxicity. And right now, all you need to know about ADCC is that you could also refer to it as activating NK cells. So there's a type of immune cell called an NK, or natural killer cell. And ADCC is a way of activating NK cells. So antibodies can activate NK cells. You can see sort of this process happening here. You can see some antigens on the surface of one of our cells, and antibodies binding to those antigens. NK cells, the natural killer cell, will have Fc receptors on their surface. And if those Fc receptors bind to the Fc portion of antibodies, then the NK cell will kill the attached cell. And so the natural killer cell will use its Fc receptors. So this is similar to what we've seen before. There's an Fc receptor, there's a cell. It will use its Fc receptors, and it will kill whatever the thing was that had antibodies on it. And so you can see this here. Both of these examples are showing this happening in the case of a tumor cell, but there are a number of different situations where this can happen. Yep, Andrew.
Are NK cells also activated to target cancer?
So both of these are showing you cancer as examples. So they can be involved with cancer, and we're going to deal with that later. In fact, some of the ways that they may be activated against cancer can involve ADCC. Right now, you just need to know antibodies can lead to ADCC, and ADCC is activating NK cells. And I promise you, later in the semester, when I ask you what ADCC is, I told you about it before. The sixth function of antibodies is, again, kind of similar to what you've seen. And it is called the activation of mast cells. So again, we've got the same idea. There's some immune cell with an Fc receptor on its surface, and that Fc receptor is binding to the Fc portion. Here, it's a mast cell, not an NK cell, and not a phagocytic cell like a neutrophil or a macrophage. And the mast cell is full of granules, toxic stuff. And when the mast cell is activated, that mast cell will secrete all of its toxic stuff. So all of those little bombs of toxic stuff will be secreted from the mast cell and be sort of shot out to kill whatever. And in fact, here you can see a mast cell full of granules, and here you can see a mast cell after it has released all of its granules. It releases a lot of its granule contents when it sees an Fc portion using its Fc receptor. And this is always sad to me because this is what happens whenever I go outside in the spring with the pollen. So those are our 6 functions of antibodies. It turns out that different isotypes are better at different functions. So some of those functions go really well with some isotypes, and some sort of go with others. So for the rest of the time today, I'm going to tell you what is unique about each different isotype. And so we're going to sort of think about various factors about different isotypes that are unique. I have to show you one other slide. And this slide comes from a lot of experience talking about all of this information. If I don't tell you this slide, then I get questions. If I do tell you this slide, then I get a different set of questions. So I'm going to tell you the slide, but I'm going to give you a little bit of a caveat. If you are a student who knows a lot of molecular biology, biochemistry, this next slide may drive you nuts. Okay? Here's the deal. Yes, there's a way, I promise it works. You're going to look at it and be like, no, that is not how this works. No, 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 no. Yes, there is a way. We're going to get there. We're just not there yet. Right now, I need you to know this is a phenomenon that exists, and trust me that there is a way to make it happen. And the answer is that antibodies can change over time. One thing that can happen over time is that they can change how tightly they bind to antigen. So antibodies made early in a response bind kind of weakly to their antigen, and over time, antibodies will bind more strongly to their antigen. This is called affinity maturation. The affinity of the antibody matures. The other thing that you should know is that antibodies can switch their constant regions. So we can have different constant regions. Here, we've got the same bacteria. At one point in the response, the antibody that responds to it is IgM, has the M constant region. Later in the response, we can switch to having antibodies with an IgG constant region, because perhaps IgG lets us do the best function. So by switching from IgM to IgG, we can get the better function that we want. So note that, yes, you could pair any of these types of isotypes, any of these constant regions, with any variable region, the ability to bind to any antigen. And that is referred to as class switch or isotype switch. Again, I promise we're going to see how that happens, just not right now. So your textbook has two tables that talk about some of the unique features of different isotypes. I'm first just going to show you those two tables to show you kind of what things to be looking for, and then we'll actually zoom in on individual isotypes. So one part of this is that we can see, which heavy chain we have. I already told you about that. We can see our different isotypes, and they do break down the different IgGs and IgAs here. But you can also see molecular mass, you can see the amount in serum, and you can see the half-life in serum, and see that there is some variation. You can also see that there's some variation in terms of the functions that we've talked about, okay? So the first isotype that we're going to talk about is IgM. And when you look at IgM, there should be something that really stands out about IgM compared to the other isotypes. So what seems really unique here about IgM? Yeah, Emma. The molecular mass is way bigger than all the other ones. You can see it's kind of like medium-ish half-life, medium-ish here, like kind of medium in a lot of ways, but in terms of size, it is gigantic. The reason for this is that IgM is generally found as a pentamer. So we don't find one IgM antibody by itself very often. There is a little protein called the J chain. The J is for joining. Don't really care. But there's a little protein called the J chain that holds together 5 IgMs. And they should be 5 identical IgMs. Like 5 copies of the same IgM. And so IgM actually floats around your body looking like this, as a pentamer. So it doesn't have 2 binding sites for antigen. It has 10. And you can actually see some electron microscopy figures of IgM pentamers in the body. And so this is one of the defining features that when I think of IgM, I immediately think pentamer. And the fact that IgM is a pentamer also helps it do one function really well. And so it kind of helps this have its standout function. First of all, let's think for a second, what is useful about being a pentamer? There are really kind of two parts of this that are useful. What could you imagine is a pro of being a pentamer here? Yeah. There are more binding sites. What does more binding sites give you? What is it? Stronger binding. You get stronger binding because you don't have avidity of 2x, you have avidity of 10x, right? So IgM has great avidity. But IgM also is really an all-star at one function. When you look at this table, what function should you associate with IgM based on this table? Yeah, Andrew. Activation of the complement system. IgM is the all-star in terms of activating complement. Yes, some other ones kinda can, but again, if I think about complement, I only ever think about IgM. What was the first step of activating complement in the classical pathway? What was the first thing that happened when we activated complement with the classical pathway? There was an antibody, because obviously we're talking about antibodies. Then what happened? Yeah, Emma. Mm-hmm. So C1 binds and has a conformational change. Do you remember what C1 looks like? I see lots of hand motions. Do you remember how I described it? An upside down bouquet of flowers, right? It has a really specific structure. Well, if you've got 5 FCs together, those 5 FCs make like the perfect binding site for that little flower bouquet. And so the pentameric IgM is the perfect thing for C1 to bind. C1 can bind to other antibodies, but then it kind of has to be like a just right geometry thing. If there's IgM around, it's perfect, and it fits perfectly. So pentameric IgM is the all-star of fixing complement. I'm not going to harp on this a ton right now, though it is something that we're going to think about a lot more later. We are eventually going to think about the order of when different types of isotypes are made, because they're made with sort of different timing in their response. And when we do that, we're going to be really happy that we have a mnemonic to help us. That's the mnemonic I already told you. Based on that, what do you know about IgM, like when IgM is produced in a response? It's first. It's the first one. So the other thing that I often think about with IgM is it is the one that's made first. And again, that's going to become a big deal later, but just for now, note IgM is the one that's made first. Okay, now we're going to move to another one, isotype. It always feels like this one's in a weird spot, but I did put it in this spot for a reason. So the next one we're actually going to think about is IgE. So now we're all the way down at the end. What do you notice that stands out about IgE? Yeah, Alexis. Serum level. What's up with its serum level?
I didn't realize that it was a negative. I just saw the 5 times.
Okay.
It's a negative and everything else is a number.
Right.
From 0 to 10.
Yeah. So it's way small. It's tiny, 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 tiny compared to all the other ones. There's way, way less of it, right? Turns out there's a reason why there's way less of it. And the reason why there's way less of it is because it doesn't hang out in the serum. It hangs out somewhere else. So all the other ones can be found pretty easily floating around in the serum. IgE is not really in the serum. And when we think about IgE, we think about one key function of IgE. What is the function of IgE? Yeah, Kyra. Sensitization of mast cells, exactly. So IgE, I told you about this function of antibodies working on mast cells. Really, IgE is the only one that does that mast cell function. And that's IgE's like starring role. So I said that IgE is not really found in serum. That's because IgE actually floats around your body and binds to your mast cells. And so all of your IgE right now is stuck to mast cells. So that if any antigen, any allergy thing, any antigen that binds it comes around, you get a response like that. So your IgE is not sitting in the serum. It's all sitting on mast cells. Mast cells have an Fc receptor that is specifically the Fc epsilon receptor that binds to IgE. And IgE is going to be able to activate those mast cells right away. So that's sort of the unique feature of IgE. I put IgE here because I want to be able to draw a comparison with another isotype. I'm going to show you first the same kind of table I've been showing you about isotypes, but it's a table from an older edition of a textbook. Okay? So this is a different version of a textbook table about IgD. And to be honest with you, this is even more up to date than I have some other ones that are even more out of date than this. What do you notice about IgD? I actually don't have the most out of this. I thought I had the more out of date one than this. If I had the totally out of date one here, and we looked at the functions, it would say minus for everything. What we used to say about IgD is that it had no known function. That had absolutely no function at all. And we would, in fact, say the only reason why anyone cares about IgD is because it's made on B cells at a certain time in their lives. And so sometimes immunologists use IgD to help them track where B cells are in their development. Because if they're making IgD, it tells us they're at a certain time of their life. And so here you can see this version from your textbook where at a certain point in their life, B cells make IgD. And so we use IgD on their surface as like a marker of their development. But for most of my time as an immunologist, we would say IgD had no known function. It did nothing. We have since discovered a function for IgD. And so this is actually the table from your textbook about IgD, and it talks about IgD. So what is the function of IgD according to this textbook? According to your textbook, according to current immunology? Yeah, Josh.
It seems to be activating basophils.
It seems to be activating basophils, and that's sort of the unique function. Do you remember back to when we talked about cells and organs of the immune system? Basophils were the super, super, super rare cells that you have almost none of. Because they're so rare, we can't really study them, and so we don't know a whole heck of a lot about them, but it looks like maybe IgD actually turns on basophils, and we didn't really know that because we couldn't really study basophils. In the end, what we think happens with basophils is that it ends up looking exactly like mast cells. It's just it's IgD in basophils, IgE in mast cells, but it's the same degranulation. It's the same Fc receptor business. We think the basophil IgD thing is the same as the mast cell IgE thing. But again, we're not crazy sure of all those details, and that's relatively new information. And so I tell you about IgD here to contrast with IgE, since you just heard IgE. Yes, Emma.
So I remember when we talked about basophils, they're typically seen also in the setting of a parasitic infection. So individuals who have a parasitic infection, would they have elevated levels of IgE?
Potentially, yeah. So that's our IgD situation. So I'm going to at least get to start. We've got two more isotypes. We've got A and G. I can at least start A today. We'll finish the rest of them on Monday before getting into the diversity stuff. So one thing that you can already see here is that IgA has two different subtypes. We're not going to really get super focused on the two different subtypes. And you'll notice that in some ways IgA is kind of average seeming from many of these parameters. But IgA does really have one thing that is super important about it. You can see that it is very good at neutralization, but it's not the only one. That's not like a special IgA thing. But it does have one thing that it is super, super important about. And that is that IgA can be transported across epithelial cell layers. IgA is found in different parts of your body than are the other isotypes. It does neutralization there, but I mean, like, we're less excited about the neutralization and more excited about the where it's doing the neutralization. You can also note that it lists here this transport as happening as a dimer. Most of the time when we think about IgA, we see IgA as a dimer. Again, there's also a J chain that holds together two IgA molecules so that we have dimeric IgA looking like this. I will admit to you, most all of my knowledge about IgA is about dimeric IgA. I have a friend who works on IgA, who I podcast with, and every so often she'll be like, but what about the single IgA? And I'm like, I don't know anything about that. But she does, and she thinks it's very important. But I'm not going to tell you anything about it because I don't know that much about it. And when we have this dimeric IgA, it's sometimes referred to as secretory IgA. Secretory IgA can bind to a special receptor on epithelial cells and actually get moved across those cells. So the cells can sort of pick up the IgA and deliver it to the other side. And so if, for example, this is the bloodstream next to the intestine, the IgA can get carried across the intestine into the open space of the intestine, like where the food is, and into the open space of the lungs, and into the open space of the nasal cavities, into those barrier tissues where we're first going to see microbes. Those areas are known as mucosal areas. And so IgA can actually move across epithelial cell layers and move into mucosal areas. So like your saliva, when you blow your nose, the snot, all of that only has IgA in it because IgA has gotten out of the bloodstream across cell layers. And that process is called transcytosis. But the point is that IgA is protecting you out in that barrier area. And so you can see this here. So this is actually in the GI tract. And ideally, we've got a bunch of IgA in the GI tract. It can bind bacteria and neutralize them so they never even can get into the GI tract. So like there, it is sort of this first line of defense at the barrier tissues. There might be a situation where someone does not have antibodies, and then we're going to have a bigger issue because we're not going to have protection at the barrier site. And so you can see IgA is in different locations in all of these mucosal sites, like the lung and the eye and the nasal tract and the GI tract, in different places than others, which are throughout the blood. Also, notably, IgA is also found in breast milk, and so IgA is transferred via breastfeeding. I'm going to have one other really interesting thing about IgA that I'm going to start with, and then we'll move into IgG, and then go into antibody diversity on Monday. So I will see you guys then. No problem. Thank you.